This is going to be a redefinition of our place in the cosmos, and that will happen if we discover life on Mars.
Welcome to the continuing series of the award-winning Mars documentary titled Blue Planet Red. There's more to Mars than you might think. Water, life, pyramids, UFOs, cataclysm. Watch the film online at blueplanetred.net. Today, we're visiting the Agnostic Life Finding Association, otherwise known as ALFA Mars, a group of scientists and engineers aiming to determine whether indigenous life is present today on Mars before humans walk on its surface. The instrument they're developing, called the Agnostic Life Finder, is a supercharged, mic drop, end-all be-all, no-holds-barred life detection experiment that will definitively determine, once and for all, whether or not the surface of Mars contains any microbial life. ALFA Mars is located at the Foundation for Applied Molecular Evolution in Alachua, Florida. Joining us for this interview is Dr. Jan Špaček and Chris Temby from ALFA Mars. Later, we'll also be hearing from their colleague, Dr. Elisa Biondi. But first, we begin this episode with Dr. Stephen Benner, an award-winning biochemist who has published hundreds of research papers in the fields of chemistry and synthetic biology. In a previous interview for this series, Dr. Benner explained how the 3 life test experiments aboard NASA's Viking mission were all consistent with autotrophic microbial respiration. In other words, NASA likely found microbial life on the surface of Mars in 1976. Today, he explains an alternative method for detecting microbial life, not through the metabolic activity of microbes, but by detecting and identifying what's known as polyelectrolytes within DNA, known or unknown to science.
For those viewers who are wondering what the dinosaur head is over your shoulder, can you please explain what that is?
Yeah, well, that's a brontothere.
Brontothere.
So you're familiar with the Brontosaurus?
Yes.
Bronto, meaning thunder lizard, saurus being lizard. So this is so there, T-H-E-R-E, is not a lizard, it's a mammal. So this is actually not a dinosaur, it's a mammal, but it's a thunder mammal, which, as you can see from its size, was a rather large guy. He's most closely related to the modern rhinoceros. And he lived in the Eocene, which is about 40 million years ago. And these were herbivores. This guy is coming, he had the misfortune of being caught in a flash flood in what is now the northwestern panhandle of Nebraska, and then being dug out by somebody who mines private land for...
That's not a replication.
That is a real fossil from the panhandle, yes.
Let's get to a central point with regard to the design of this Agnostic Life Finder device, okay? We call it agnostic because, as I understand it, it is designed to look for microbial life as we know it and as we don't know it, and that is because of your work in the field of synthetic biochemistry. And there is something called the polyelectrolyte theory of the gene that is at the core of all this. Can you explain what that means?
I can try. So yes, Darwinian evolution is believed to be the only way that organic material can organize itself to give the properties that we value in life. And further, we are under the impression that information is important to Darwinian evolution. The word, as we say in John the First, and the word must be made flesh. It must be made into a molecule that can be copied, that can have its structure change, and that the structure changes can themselves be copied. And so it turns out that's a difficult set of properties to get in any molecular systems. It's not present in proteins, for example. If I change one amino acid in a protein, it can cause the protein to precipitate. It can cause the protein to unfold. It can cause the protein to dramatically fail at performance. And I cannot have an informational molecule, a gene, so that if I change a piece of information in that molecule, all of a sudden the molecule precipitates, right? It won't work. So what happened was many of us and other laboratories, Pete Schultz at Scripps, Floyd Romelsberg, lots of people, went out to try to design alternative informational molecules that could support Darwinian evolution. Maybe 300, 400 of them have been made. Some of them work, some of them don't work. And from that, the ones that work, you come up with general structural features that explain why they work.
So you're saying a structure that looks similar to, but is different than the DNA double helix structure that we're familiar with. It's like that, but different.
That's right. And we discovered that we can change the bases in the DNA, which you learned about in 10th grade biology. That is the information components, GACT, the 4 letters of DNA that we use to encode information. We can change the sugars in the backbone of the DNA, but what we cannot change is the repeating charges that hold the building blocks together. And so this is an electrolyte. I mean, this is something that you know of from the point of view of a salt, right? So you worry about your electrolyte balance when you hike into the Grand Canyon and it's 95, 100 degrees centigrade, Fahrenheit, and you worry about your electrolytes because you're sweating them. But what DNA is a polyelectrolyte. So the white things in the background are phosphates on the backbone. So these are the repeating charges in the backbone of the DNA. It turns out that these are the features of that structure that allow this molecule to evolve. Allow, more precisely, its structure to change so that the information it encodes can change. And it's the change in that information that does not disrupt the behavior of the molecule, does not call it to precipitate. I can change now the information content, which is the various colored things which you can see in the backbone is here. The bases are in the middle. The bases can change to change the information that's encoded in this molecule without changing the backbone, which still has a polyelectrolyte theory. So even if you change the structural information in this molecule by changing the structures of the things in the middle, the molecule still does not precipitate, it does not explode, it has pretty much the same reactivity, and it can continue to evolve even though the information in it changed.
You guys are actually cooking up this stuff for real here in your lab here, okay?
In fact, this is a 12-letter DNA molecule. If you look closely...
Versus a DNA is a what?
Well, you have 4 letters
4
in your
letters,
DNA.
okay, so our DNA is 4 letters. This is a 12-letter DNA, which you can make here.
Yes.
You've made it.
We have made it.
This is not theory.
This is not theory.
Okay, what is the theory is how evolution can occur. Is that
right.
what you're suggesting?
Exactly.
Is that based on...
So I've got an orange and a blue, and I've got a green and a yellow. That's information. Suppose I wanted to replace this green and yellow by an orange and a blue. That would change the information. It would be a mutation. The backbone polyelectrolyte so dominates the physical properties of this molecule is I can exchange red, blue, orange, and so on, and still have this molecule form a double helix, still be replicable, still dissolve in water. So what Chris Temby and Jan Špaček and the Agnostic Life Finder are doing with NASA support, NIAC, it's an innovative concept of program, is trying to build a device that will not detect the colors, because they can be different in the middle, but to detect the polyelectrolyte in the backbone, because the polyelectrolyte is universal in all informational genetic molecules to support Darwinian evolution. And we think that Darwinian evolution is universal to life. Well, we don't think it. Everybody thinks that Darwinian evolution is universal. So what we're looking for is not a molecule that is made by Darwinian evolution or organisms that have evolved, because a lot of those molecules can be made also by non-biological processes. What we're looking for is the molecules that are essential to enable Darwinian evolution. Those are these polyelectrolytes.
So you're saying that maybe there's DNA like this on Mars.
Yes.
That's
12 letters.
Okay, and this Agnostic Life Finder could detect that.
So if Elon Musk puts SpaceX down on the surface of Mars and he's going to pump water from the subsurface as a resource to drink for the astronauts to drink, but also to make propellant to bring the astronauts home, we can put a device astride that flow of water, a huge astrobiology sample, and pull out polyelectrolytes, concentrate them, and then detect them and to determine whether they have other properties that are essential to support Darwinian evolution.
How did you figure this out?
30 years of making alternatives for them, some of which worked and some of which didn't, where we discovered that the polyelectrolyte alternative to DNA worked, and the ones that we got rid of the charges in the backbone did not work.
What made you think that you could just come up with new DNA?
Well, the funding agencies didn't agree with that, so we moved to Switzerland to do it because we couldn't get anybody in the United States to agree to fund it, and the Swiss were crazy.
But you must have thought that it was going to work.
Well, we thought it might work. Yes. Well, again, I mean, we were Henry Hudson going to look for the Northwest Passage, right? We certainly didn't start out to this project to rebuild biology from the ground up, right? We didn't expect that we were going to fail 100%. We may have been a little bit more optimistic than we should have been given the 30 years that we have put into this project.
This is pretty cool stuff, man.
Well, thank you. Yes, I should talk to my funding agencies.
Well, okay, so let's talk about funding. Who can we talk to?
Well, so the National Institutes of Health. So this is in a product which Mickey Urdia and Chiron and Bayer Diagnostics put in to monitor the viral load of patients infected with HIV or hepatitis C. So if you have had hepatitis C, it's quite possible that your viral load was measured by a DNA molecule that we built with 6 different building blocks instead of the 4 found in natural DNA. We have a major project right now with the Department of Defense to detect, pathogens in your blood. If you have sepsis, that is blood poisoning, we have a tool using some of these alternative DNA forms to find them and to let your caregiver know what antibiotics to prescribe and what not to prescribe. So there is, in fact, some of the royalties for licenses that we collect on these inventions are paying for the building that you're having this interview in. So yeah, there's a lot of practical value of having a kind of DNA that is not like you and me, but can do things sort of like what our DNA does, but it can do it differently and orthogonally, and it turns out to be very useful in building diagnostics process. And then we're evolving a whole line of therapy based on the fact that this kind of DNA can evolve. And because it has more information density, it can evolve to give you more useful things than the evolution of just 4-letter DNA can. So there are compounds made out of 6-letter DNA that bind to cancer cells, that deliver drugs to cancer cells, that pull down targets from cancer cells. And you can talk to the National Cancer Institute because the peers there have been very slow to understand the potential of this.
Could we interest the military in funding this somehow?
So as I said, the Department of Defense is already funding it. So yes, I mean...
But we needed to get it to Mars.
Getting it to Mars is a good question. I think that would cross institutional boundaries and have a turf war. I don't think that NASA is required to do the Mars stuff.
DOD did Clementine.
That's a good point, yes.
They got to get this thing Mission Impossible style onto Mars. How far did you get with the book last night? Were you able to work on it?
I did not finish it, yes.
Okay.
But I did not finish it. I spent too much time reading the paper from your
You were kind enough to share with me an advance copy a few months ago.
Certainly it's a number of chapters. We are, of course, this book has gone a lot further and a lot longer in the last couple of months since we communicated last.
Yeah, you've been working on it. But the version that I read starts with chapter 1. NASA is not looking for life living today on Mars.
That's right. That's right.
That's pretty bold first chapter.
Well, it's sort of just a statement of fact. I mean, NASA, and that's sort of been a policy decision from NASA, as the chapter goes on to say. There was, of course, an extant life living today mission in 1976. What happened was it failed to produce unambiguous results, and to some people's minds it showed that life was actually not present on the surface of Mars. The immediate response was the mission closed down, Mars exploration stopped for a quarter century, and so when it started up again as a result of the formation of the NASA Astrobiology Institute, which itself was the result of their having discovered cell-like structures in a meteorite that came from Mars by way of Antarctica, NASA decided that it would not look for life explicitly, but it would look for habitability, it would follow the water. These were the mantras that were guiding the missions. And so even today, if you look at the rovers on the surface of Mars, they're looking for past habitability. not current inhabited life forms.
Well, so let's pause right here, because if you were to say to somebody out on the street, NASA's not looking for life on Mars, and they say, oh, of course they are. They keep sending all these missions there, and getting to this point you were just raising, well, they've adjusted over the years. They are in fact looking for signs of ancient life.
That's right. Or ancient habitability, which is a statement not about life exactly, but a statement about the environment, so as to assess whether it was possible in that environment for life to exist. So habitability is a indirect way of saying we're looking for the possibility that there may have been life at some point in the past. And so Gale Crater, where they're currently looking, the rovers are walking around looking for things, are in places that are specifically designed actually to not be the places where life would be most likely to exist if it existed today on the surface of Mars. So there are what are called special regions. Perhaps you have talked to other people about this. These are places where water might be present near the surface, where life might be present. And there's a concern about what they call planetary protection. There's a concern about going to those places because there's a concern that if we go there and we bring life from Earth, it might settle down in the special regions and outcompete indigenous life. So right now, Mars missions are specifically designed to not go looking for extant life in the places where they're most likely to be found. Now this is evolving. A lot of us, Carol Stoker is an example, but certainly myself, we have been agitating in the NASA community. They really should be going looking for extant life. So in 2019, Michael Meyer set up a meeting in Carlsbad, New Mexico, where 60 of us got together and said, this is where life should be sought on Mars living today. And there's a whole conference document that came out of that. In 2023, the decadal survey, every 10 years, NASA goes back and tries to come up with a plan for going forward for 10 years. This was last done a year ago. And Amy Williams over here at the University of Florida did put together a team that assessed the possibility of looking for extant life on Mars. We're talking about 2042 is when they're thinking about looking for life on Mars. So you will still be here, Brian. I might not be, but the point is they're not looking for extant life today. Almost all of the resources that have been committed to Mars as a planet are on not finding life. The overwhelming majority of those resources right now are trying to get samples returned from Mars. But that is itself a decadal-long thing. We're not going to have samples back from Mars in a year, 2, 3, or 4 years as the way things are going. And even when we bring them back, they will not be samples relevant to the search for extant life, that is life living right now.
And you say, reverse engineering their thinking, this, get ready to start getting ready.
Yes.
To address a question of secondary importance rather than addressing a question of primary importance.
That's right. So the book, one of the reasons why I wrote this book was to try to help the layperson understand the thinking that goes into those kinds of mission decisions. So there's a reason why NASA is approaching Mars the way it is approaching Mars. A lot of that reason goes back to the 1976 experience, where NASA regards that experience as having been a failure at some level. It was not really a failure. Actually, we think that the experiments overall were quite valuable and should have been followed up on, but that's not how the culture has come to view the Martian results from Viking. And that led to a hiatus for 25 years. Essentially, nothing was sent to Mars to do anything. Now, our view, of course, is that it's quite clear now that the results were misinterpreted. And so there's been a long back and forth between the people who say, wait a minute, the results are misinterpreted, and Martian mission planning should reflect that fact. And the community, which has not really found any of the data that don't internalize the results of those data, don't actually change their community view, because it's in textbooks, it's very tightly right now, culture is hard to change, and we'll have a discussion perhaps about culture. But yes, the purpose of the book is to let the public understand exactly the thinking that goes into Martian mission planning, and why, for example, 2 years ago, NASA set up what they're called interdisciplinary centers for astrobiology research. ICAR is the acronym. And most of those centers were funded not to look for life on the solar system, on Mars for sure.
We'll get to that.
But rather to do things with the NASA basic research program that were not actually relevant to this question at all.
What you're unfortunately highlighting for the layperson is that, again, getting back to your book, not only is NASA not looking for extant life, it's not even looking for past life with any particular urgency.
There is becoming an urgency because various entities, NASA, the Chinese National Space Agency, SpaceX, Elon Musk, these people are promising to send humans to Mars in 2 more launch cycles. Depends on what happens when they will arrive, but it's within years, not centuries. And so the idea here is that once humans arrive, the plan to search for extant life is going to change, because you are going to bring Matt Damon and you're going to bring his poop and you're going to grow potatoes in it. And so there will be an issue of how to manage the search for indigenous life once human life is on the surface.
It seems like it just got so confusing over the years that people are just tired of it.
Yes, well.
And here we have you scientists down here in Florida who are trying to take chest paddles to this whole issue and revive it, perhaps even single-handedly among the the academic and institutional crowd. But okay, let's just say we go up there and this new gizmo of yours, it does in fact find Martian life, whether it be like our DNA or alien DNA. So what?
Chris and I are writing a blog post with the title, "Read the Goddamn Paper." Okay, that's a common statement, and we're interested in pedagogy.
I hope you title it that for real.
Well, we'll try, but the point is that it's pedagogy, right? We're teaching students. This is an intergenerational exploration activity. It's like Columbus didn't go to the New World without the generations that would follow him. And Leif Erikson, of course, failed to discover the New World because he neglected to have generations that followed him. That's the only difference. So we have to persuade students to go back and actually read the papers. It's like we train lawyers. We don't train them to read the cases. We don't train them to read what people say about the law. We say, read the law. Suppose we find life on Mars. How are we going to announce it to the public? NASA is worried about this. They say this is going to be an administrative level decision, which means that the President of the United States will announce that finding. Just like Bill Clinton in 1996 went on to the Rose Garden to announce the very tentative observation of cell-like structures in Martian meteorites. So this is, if they announce a cure for cancer, they're not going to have the President of the United States out there on the podium, right? This is going to be a redefinition of our place in the cosmos. It's going to create wonder, astonishment, fear. There's going to be all sorts of things going on in the public sector. And so even NASA will tell you in writing, this is something that they have said, that this will be an administration level decision. If you want to go to science fiction, which I prefer not to do, right? What happened when they first contact, when, Zephram Cochrane first discovered Vulcans? Well, the answer is the whole world changed. Peace was achieved in a decade in the utopia of Star Trek, which is famous to your viewers. Not the dystopia of Star Wars, which of course is a much less promising view of a future, but fortunately that's in a galaxy far, far away, right? This is going to be a fundamental transformation as big as, for example, Columbus discovering the New World. It's a whole perspective that changes when you make a discovery of this magnitude, and that will happen if we discover life on Mars.
Quoting from your book, one thing is clear: you want to know if alien life exists out there in our solar system today, not in 20 years. Mars is most likely the place to find it. Not Europa, not Titan, and certainly not on planets orbiting other stars light years distant.
What is clear is that we have today access to Mars. It happens to be conveniently the most accessible planet. Venus might be also considered to be accessible. But Martian life, the geology of Mars, we understand, the chemistry that you spoke to Elisa Biondi about, which is showing how life may have originated on Earth, could also have occurred on Mars. It may have occurred on Venus. The planetary evolution on Venus, Earth, and Mars has been severe in all three cases, but Mars, it's quite possible for the life that we know of could have survived the planetary evolution. Mars is the only location where you're going to be able to, in any human timescale that is in the next century, establish the presence of life.
It's almost like moving the goalpost.
It's moving the goalpost in the sense that if you are concerned, if you can't tell me whether life is on Mars right now based on experiments where you've landed stuff on the planet, you've gotten rocks back from the planet, you have all had 4 generations of rovers on the planet, you're certainly not going to be able to do that from astronomical observations, which are limited to photons of light coming from the exoplanet. You're certainly not going to be able to come up with a definitive case. It won't advance us anywhere towards the possibility of knowing whether those planets host life or not. And that's the question that the public wants to know an answer to. So the President of the United States is not going to go to the Rose Garden if we discover a rocky-like planet circling, a star 18 light years from us, right? But they will be going to the Rose Garden if they discover life on Mars.
Dr. Jan Špaček earned his Ph.D. in genomics and proteomics from Masaryk University in the Czech Republic in 2018. He's currently a molecular biologist, analytical and organic chemist, and senior research scientist at the Foundation for Applied Molecular Evolution. Dr. Špaček is also the CEO of ALFA Mars, as well as the inventor and lead designer of the Agnostic Life Finder instrument.
Given the history of NASA's search for life on Mars, some would argue they're not doing enough, that they're not sending the instruments necessary to actually look for and discover life on Mars. So because NASA won't, you guys will.
Since Viking, which was 1976, we have not sent a probe or instrument to Mars which would be able to detect life if it was sparse, as we have it in, for example, Atacama Desert in some regions. So the instruments we are sending to look for extant life are not sensitive enough to actually find it. So that's the issue. And yeah, NASA is not doing anything in that regard.
What we're talking about here is extraterrestrial. And we don't know for certain whether or not extraterrestrial, even just microbial life, if extraterrestrial microbial life looks anything like what we know as life here on Earth, terrestrial. So why even bother if we don't even know what we're looking for?
There's a lot of scientists who believe that life on Mars, extant life on Mars, is quite likely. I'm one of them, and I believe that before we send humans there, we should know if it's there or not and study it if possible.
The problem is, within the astrobiology community, you said there's a lot of scientists who do believe there's life there.
Yes.
In my experience, there's also a lot of scientists who think we shouldn't bother because we don't know what we're looking for. Now, I understand that is the nature of what you're trying to accomplish with the Agnostic Life Finder, but tell us why the skeptics, why the people who don't think we should even bother in the first place, tell us why that they're wrong.
It's not that they are evil people or something. It's just, hey, we sent Curiosity and Perseverance to Mars, and now it's time to fund our telescopes, right? So I think that's the thinking why search for life on Mars is being pushed, like diminished.
But what I'm saying is, there are people who think we don't know how to look for life as we don't know it. But you have a different perspective. You think that we can look for life as we don't know it and are in fact building something to do just that.
Yes.
So if someone's challenging you at a conference, Dr. Špaček, we don't know how to do this. What makes you think we can go do this?
Basically, you are looking for polyelectrolytes that carry informational building blocks that allow basically transmission of genetic information between the generations. And this point is not contested at all. Everybody agrees. It's just,
who's everybody?
Well, everybody on the conferences, when you, or I would say this, nobody disagrees that this would work. The contentious point is that there's not as much of DNA in your sample as other things, so you actually need to sample larger sample sizes. So from that perspective, it's a different way to do astrobiology than what was the common approach. So normal approach is to use very small samples, in which case if there's just few nanograms of DNA, or even worse, some alien genetic polymer, you would miss it because you would need extremely sensitive detection instruments, right? To detect nanograms of something with a certainty, it's quite impossible if you are not looking, if you don't know what you are looking for. So the main point, how to make this work, you need to have huge samples and concentrate from those huge samples, especially if we are looking for something which is not DNA. So the contentious point is that if you are looking for alien genetic polymers, they would be very low concentration, and so you might miss them. It's not contentious point about that aliens would not use some DNA or alien genetic polymer. So, and we have solution for that.
Perhaps in your circles, you find this point not to be in contention. How much are you on Twitter?
Not at all.
What I've found is that there's very smart people, just like you with PhDs. They have lab coats too. All right. And rooted in their instruction, rooted in their study throughout their academic journey, is this narrative that we don't know how to look for alien life. Mm-hmm. I find this to be quite common with the people that I encounter upon doing this little project of mine. So for those people who haven't yet heard of this, who don't go to the same conferences you go to, simply, what do they need to understand about the possibility of detecting alien life?
We call our ALFA, the instrument we have, Agnostic Life Finder, agnostic, in a sense that we don't carry any specific belief about the life which we are looking for. But there might be some people that say, "Oh, you would not find this specific, made-up life form." Right? It's not perfect, but it very likely covers. It definitely covers all life as we know it, and it would cover large possible spaces of life as we don't know it.
Okay, so for those people who don't have any idea what you just said, the answer is yes. It is possible to look for life that we don't know about, that is not familiar to us here on Earth.
Yeah.
Yes.
Yes.
You wouldn't be building this thing if you didn't think it was possible.
And yeah, we can definitely do that. So.
All right, fine. Dr. Špaček, you think you can go to Mars and look for life? Why do we got to do that right now? Why can't we wait? Put that off to the future. Why right now?
You want to study life on Mars before you send humans there because it's always better to know than not to know. Like I watched some sci-fi Alien where the group of scientists went to a planet and there were some spores and they breathed it in. I would just switch it off because, come on, scientists would not be that stupid to go on a planet where can be some alien
Great
life.
Soundtrack, terrible premise.
And NASA is now currently doing exactly that. They are planning to send humans to Mars without knowing, with actually expecting life to be there, but without studying it before we go. So I don't think that there could be the direct danger from Martian life to humans because that's with parasites, you need a co-evolution. So, but there are different reasons. The introductions of new species to new habitats can be problematic, as you know, with pythons in Florida, but it can be on bacterial level. So
you guys wouldn't be seriously working on this if you didn't think there was a good chance we'd actually find microbes there, right?
Yep. Large portion of astrobiologists actually agree with this view. It's just that the part of astrobiologists who prefers to look for life elsewhere in the universe are better in securing the funding.
I was watching one of these lectures that you sent me recently. Well-dressed guy up on stage talking to students, okay? My question to you is, when you're presenting this information, like you're doing to me now, about your work on the Agnostic Life Finding device, right? And discussing the history of NASA's search for microbial life on Mars, and you're trying to make the case that we should go back and, let's just say, confirm, okay, some results, if we're looking at these results favorably. As it relates to the students themselves, do you get the sense that they understand what you're saying, or do they think it's science fiction?
And it's a big surprise to students. Everybody thinks, hey, NASA is doing the best to find life on Mars, right? They would not just like leave it be. That it's something to take in, and by the time they take it in, I'm already gone. So, and then some people, of course, messaging me or us as a ALFA Mars team, and we discuss it further. But
is it the first they've heard of this?
I think in many cases, yes.
Have they heard of Viking before?
I think if they did, they heard about the textbook interpretation, or the interpretation you can find in like the old texts, that the results were negative. But we cannot hold that view anymore, because the view that Viking results were negative is based on the mass spectrometry results showing that they could not detect any organic material.
The new interpretation of these results, you know, in the decades since, they've only heard the original story. They haven't heard the evolution of the story.
That's correct. Given among professional astrobiologists, the view that Viking was negative, provided negative result only, it's quite prevalent, surprisingly.
When they ask questions to you, do you get the sense that they're still caught in the weeds of the technical application of the device, and, you know, polyelectrolytes and solid stain nanopores, or do you think that some of them actually examine this information more from a philosophical, you know, how the implications for finding extraterrestrial life and what it means philosophically?
Again, I think it's more pragmatic. The people who take time to study the Viking experiments in any depth, or listen to you when you are talking to them, they are already on your side, and people who are not interested, they are not going to spend the time to actually study it. So
for the people who take a favorable position, okay, who are, you know, who are listening to what you're having to say and not disagreeing with it, you know, are they so focused on the nitty-gritty details, or do you think some of them are inspired and see the bigger picture here, or rather examine this from a different, or less scientific, less engineering point of view, and a more general curiosity, you know, wonder of like the early childhood wonder that we have that gets us into these professions in the first place?
Yeah, I think it's both. You end up in this profession because of the wonder, because you want to find, you want to know if there's life on Mars, right? And these deep philosophical things, but you get this far, so that what's get you on a journey. But to stay on the journey, you got to be a little nerdy, so you get into the details anyway.
Can you please describe basically for the layperson how such a device works? You propose to send a little device here. And that's not a robot, but it's a little mechanical device or something, to Mars. How we get it there, that's a different problem. But once it gets there, you're gonna plop it down. What's it gonna do?
So to understand, if you send instrument that is capable to take large quantities of water, which will be mined for in situ research utilization for future Mars colonists, let's say, or visitors, or to refuel rockets. If you take that water and extract all polyelectrolytes, those are those genetic molecules, then you have a good chance to either find indigenous life, or with a high degree of certainty say there is currently no life on the surface, or there has not been life on Mars surface in the last couple of million years.
So you want to send a little box to Mars. You want to mine out the ice and minerals from the layers of deposits over the past 100,000 years. You want to then put in enough quantity of water and minerals through this device that you're building.
Yes.
And this thing's gonna more or less filter it out a little in a fancy special way.
Yep.
And this little box is gonna be able to tell whether or not there's microbes, active living microbes,
so
living in there?
Fancy filtration in this case, it's electrodialysis. Filtration is
electrodialysis.
Yes, electrodialysis. That's the term. So you are using electric field. You have plus and minus, and of course, negatively charged molecules go towards positive electrode. Positively charged goes to negative electrode. To not go into details. So that's the instrument. What it does. It's a just concentration device. And then we are making streams of a liquid which contains those polyelectrolytes, genetic molecules, which can be million-fold concentrated as opposed to how you find them normally in ice. So you go from 1 metric ton of water ice, you go to 1 milliliter. So if there is any bacteria in there, it would be in its genetic polymer would be in your tiny sample, which you can then analyze with mass spectrometry or using nanopore sequencing or solid state nanopore or other methods. So our instrument, the crucial part what we are doing is taking those large quantities of water and turning it into manageable size samples, which can be then analyzed, and then those instruments will tell you if there is life or not.
Okay, so you want to take a large volume of water out of the soil. You want to then filter that down to get a good sample of that water, which you think there's a high probability of finding microbes in there, because you need a large sample because it could be diluted, you're saying, right?
Right.
So you want to boil it down to, you want to, not boil, but you want to essentially get the best sample. Concentrate, thank you. To then use that sample, that concentrated sample of water, you feed it through this new device you're building, and it's going to...
No, yeah.
No?
You overcomplicate it now.
Oh, I'm overcomplicated. Go ahead.
The concentrate is the product we are making with the ALFA. So all the genetic polymers which were originally in that huge sample are now concentrated. So if there were few bacteria originally, you would probably miss them if you just sampled, some small samples. But because we take it all, scrum all the DNA into tiny sample, then you can find the DNA in there or positively charged polymers on the other side.
Positively charged polymers, meaning alien life.
Right. To be alien, you don't have to be with positive charge, but if you have positive charge in your genetic polymer, you are definitely alien.
The ALFA device, you filter enough water through it, it is able to separate out the signs of life, or life as we know it, on one side of it, and then life or signs of life that we're not familiar with on the other side. So this way, this is why you call it agnostic, because it's searching for life as we know it and life as we don't know it, and no matter what's up there, we're going to be able to find it.
I'm not going to fight you on details, but yes.
All right. And as I understand it, you guys are building a prototype right here in the lab.
Yeah. The stationary one works perfectly as it should. Separates negative DNA. We are using DNA or positively charged polymers, which we made with synthetic chemists in here representing the alien genetic molecules.
You're saying that you guys make alien DNA here, DNA that's not like what life uses here on Earth.
Well, that's Steven's specialty.
Right, yeah. You can
yes.
test this device because you're able to synthesize DNA that is unlike the DNA that is in life forms here on Earth.
Correct.
So you know this thing's working. You can filter out the synthetic DNA versus...
Right. So a lot of products in the milk industry and food industry, beverage industry actually go through these membrane electrodialysis. So they are robust methods. We know that they work. So now we are just showing that the application works. So we don't need to reinvent the wheel.
It's one thing making it in a lab, it's another preparing it to send it to a different planet.
Right.
To get it to that
right now,
phase, what would that require?
2 years to have a space-ready instrument. It's possible with good enough funding.
Is NASA aware of your work here?
This work is funded by NASA Innovative Advanced Concepts. The problem is when people think about NASA as one hive mind, right? You have different factions of NASA and they are not talking to each other. Like astrobiology is not talking to crude exploration, human exploration, and that's an issue.
So you're saying that there are parts of NASA that are aware of the pursuit of astrobiology, the study of astrobiology on Mars and other planets, who are familiar with this type of work, but there are other parts of NASA that aren't. But from the public's point of view, they don't know this. They don't understand this nuance here. So they just understand NASA, their official position is that there's no evidence for life on Mars, but we keep sending these rovers there. But it's a little bit more nuanced than that, you're saying.
Yeah, definitely. And even with regards to what public thinks, right?
So what are the chances that the more sympathetic departments of NASA, who are at least partially funding this work, what are the chances that they will be able to convince the rest of NASA that this is important enough to include on a rocket sent to Mars?
So it takes a long time if you go through the official channels. So NASA does not, well, they are careful and slow. So the problem is that we don't have the time, right? Normally, from conception to execution of astrobiology mission is about like 20 years. Now we are at the conception, so it means that maybe late 2040s, if we go through standard NASA procedures, this instrument would be ready. So that's why we are seeking the funding elsewhere as also, because...
Too much red tape.
Yeah, it takes time. Sending humans to Mars, it's a great step forward. I think it's bigger leap, much bigger, like order of magnitude bigger than sending humans to Moon, especially if it's going to be a permanent station on the Mars. I think we should do it, but before we do it, we should study or search for life, and then if we find it, study it, and then make decisions, educated decisions, without going like in that Alien movie into unknown. So that's my take on it.
Chris Temby earned his degree in physics with minors in astronomy, planetary sciences, and history from the University of California, Santa Barbara in 2023. He currently serves as the president of ALFA Mars.
The kids these days, okay, they're interested in sending humans to Mars. It's quite popular, especially on the internet. Let's get going. Let's send up some rockets. Elon Musk wants to do just that. He's probably the biggest proponent of this idea. We need boots on the ground. We need to colonize. We need Earth 2.0. Forget Mars, we're gonna rebrand it, all right? But there's more to Mars than just human colonization.
I don't think that it's as simple as let's go get boots on the ground, and I also don't think that it's as straightforward as we have a rocket, let's go do it. I think that there's a handful of prerequisites and conversations that need to be had before we're going so gung-ho about it. And now with our understanding of extremophiles and the way that life can adapt to fill many different niches that exist, it seems again now likely that there could be life there. And with that shift in understanding, there should also be a shift in priorities, or the way in which that explorer does their exploring.
Part of the skepticism with your work is that NASA hasn't found any evidence of life on Mars, yet here you're president of an organization that is specifically tasked with finding life on Mars. So why is this not a futile attempt at a new discovery?
I think that it's easy to say that NASA hasn't found any evidence of life on Mars, especially given the fact that they're not looking. So they haven't really done an experiment since 1976 to actually look for life on Mars. So if the criticism is, well, if there's no evidence for life on Mars, why would you go look for it? My blunt response would be, there is no evidence because we haven't been looking for it, so we should start looking.
Why should we be interested in finding extraterrestrial life, and why should we be specifically looking for it on Mars?
I'll start with the fact that it's really inspiring to be able to say, well, one, we're going to look for alien life. It's really an eye-opening conversation, and it brings people in from all over the world, many different walks of life. You don't have to be an astrobiologist to be curious about what's out there and the possibility of alien life, especially somewhere so close as Mars. So there's inspiration. There's a lot of scientific knowledge that we could gain, especially if that life has a different biochemistry or a natural history than life on Earth, right? If it comes completely separate, life on Mars has a completely separate origin than life on Earth, and we're able to find it and study it, I would imagine there's tons of insight that we could actually gain about life in the universe from a separate genesis. And I don't think there's any pushback about that. I think the more the pushback would be, well, is it there in the first place? And also, is it different? Like if we were able to find that there is life on Mars, but it's DNA based, or there's a lot of similarities with Earth life, then there might be some sort of controversy. Is it just contamination, or? But that's a different conversation altogether. I will also say that there's high likelihood of incredible biomedical advancements from being able to study life, with a different genesis, right? If it started differently on Mars than it did on Earth, there's huge biomedical potential there. But even if it didn't start completely differently, and it did have common origins with Earth life, but has now been exposed to the Martian environment, evolved over eons, right? There's almost guaranteed to be some sort of evolutionary advantage or niche or some sort of adaptation that Mars life would have discovered, if you will, invented, that Earth life hasn't, because of just the different selective pressures. Humans aren't going to Europa soon. Maybe, a couple centuries down the line, that would be pretty cool. But humans will be going to Mars. Unless something crazy happens, there's going to be some humans going to Mars. And if there's life living on Mars, and humans go before we are able to look for it and actually study it, then there's a higher likelihood that we won't be able to do that search and study of Martian life because there's too much Earth contamination. So that's the timeline incentive, that we should be able to do that search, not only because it's cheaper, in the long grand scheme of things with those currencies that I just talked about, but also because we're running out of time to really do that crucial search.
Chris, you are president of the ALFA Mars group, otherwise known as the Agnostic Life Finding Association. You guys are building a little box, a little device known as the Agnostic Life Finder, aka ALF.
ALF, yeah, that's the guy right there.
Okay, so what's this magic box you guys got cooking up back here in the lab?
So the Agnostic Life Finder, or ALF, can I put this down? So the Agnostic Life Finder is a small add-on instrument that will be intended to filter mined water or collected water, and filter based on size and charge of the molecules that are in the water, in order to concentrate and isolate a specific type of molecule, which we have deemed the polyelectrolyte, which we believe is the archetypal feature of an informational biopolymer, which is a molecule that is necessary for Darwinian evolution.
This little magic box you guys got is able to determine the difference between normal DNA and, let's say, foreign DNA that we've never seen before.
Yes. Every step in science, right, is an iterative process. So you do some experiment, you make observations. In that experiment, you come to a conclusion. Let's say the conclusion is we found alien life, but we haven't really fully studied it. Okay, then the next step would be now we know that we've actually found this thing, let's go study it even deeper.
Certainly some in the Mars community are sympathetic towards your cause, but not all. Mm-hmm. There are others who want to throw shade at you life on Mars people. How does that make you feel?
There's some people who think that there's no life on Mars, and then there's an argument that needs to be made with those people to be able to convince them that maybe it is possible and we should actually do that search. And then even amongst the people who do think that there could be life on Mars, there's already that common ground between us and them. However, they might say we have no idea what we're looking for, even though we've presented, the polyelectrolyte theory of the gene is an example of an agnostic biosignature that we could actually go search for, not only on Mars, but in the solar system. And some people might agree with that conclusion, and some people might not. So that's another argument that needs to be had. And then even if people agree that the polyelectrolyte is an example of agnostic biosignature, they still might not agree with the detection methods that we've put forward as, for example, the Agnostic Life Finder.
Regarding the Viking results, there have been some disconnects between the data versus our understanding.
What can't be explained is that if there is an oxidant that is so strong that it oxidizes the labeled release food within minutes, how was it possible that the pyrolytic release experiment was able to actually fix carbon over 6 days in the soil? Would that oxidant not therefore also be strong enough to burn off those the carbon that was fixed in the soil? So conceivably, the culture that came downstream from the Viking results has just kind of forgotten this nuance.
Why are you down here in Florida working with Dr. Benner?
I'm just inspired and curious by the nature of the universe, and there's so many questions out there. I've always been asking questions, even as a little kid, about what's around me and how to explain it. And I found that actually the most questions are out there, out in outer space.
Well, hang on a second.
Sure.
We got TikTok these days.
Sure. That doesn't fascinate me.
It fascinates a lot of people.
Well, I think it consumes a lot of people. I don't think that they're that fascinated by it.
You're going against the grain there.
There's a lot of man hours being spent on the black hole of the internet and the algorithms and the 5-second swipe and consume, as opposed to the larger questions and the bigger framing of it all. We need to go look for life, and there's a lot of great ways to do it, and we're just really trying to push the boundary of the most experimental ways. How can we really test what's agnostic about life in the universe? And so, the polyelectrolyte theory of the gene has informed the development of ALF, and ALF is just one way that we can look for an agnostic biosignature.
Dr. Elisa Biondi earned her Ph.D. in genetics and astrobiology from the University of Florence in Italy in 2007. She's currently a senior scientist and fellow at the Foundation for Applied Molecular Evolution. In 2022, she and her co-authors published their research demonstrating how RNA can spontaneously form when certain organic molecules, known as nucleoside triphosphates, do nothing more than percolate through basaltic glass created by lava expelled from volcanoes. This discovery provides a potential breakthrough in our understanding for how life might have emerged on Earth and Mars, where both planets had similar volcanic activity early in their formation.
Did you watch Star Trek growing up?
Nope.
Star Wars?
No.
Any science fiction?
Nope.
Yes?
No.
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 Huh.
I'm not into that.
Not into that.
Nope.
Aliens?
Not really.
Not really.
I mean, I'm fascinated by the idea of actually having, discoveries and trying to figure these things out. But when you see it in a screen or when you read it in a book, of course I read Asimov because I'm a classical person. I really liked it.
Isaac Asimov.
Yes.
Mm-hmm.
But besides that, it tends to be a bit too made up, and I actually am more into concrete science and what you can do to answer some questions and what you can't do also.
So how does this research help us look for and confirm life on Mars?
It's another proof of concept that what we're getting to is that the polyelectrolyte theory and what you should look for in general is not exactly what we have here, but it's a more flexible idea of a polymer that has the fundamental characteristics that are needed for Darwinian evolution. And our systems are actually helping us understanding what are the abilities and the features that are the same and different in polymers that do not exactly match the DNA and RNA that we have on Earth.
It's very important work. You guys are doing stuff here that's unique, that no one else is doing.
Correct.
That's pretty awesome.
I absolutely agree. I wouldn't be in Gainesville, Florida if it wasn't for a great job, right? A great gig. Yeah.
Let's talk about the origin of life.
Let's.
The way I understand it, and the way a lot of the public understands it, is that there are two competing ideas for how life started here on Earth. It spontaneously emerged, or it was brought here. This idea of panspermia, right? Seeding the cosmos and such. I understand that your work, you have some recent research that perhaps adds to this discussion about life emerging here on Earth independently.
I'm not sure, though, that those are concepts that are necessarily mutually exclusive or competing, because you can't have spontaneous appearance of molecules just by chance, by how molecules meet and react to each other with components that have been seeded and feeded from the outer space. It's all about levels, so you have to, if you want to focus on what is present in the atmosphere and on the crust at this specific time, then yes, you can decide if it's outer or if it's inner, but the truth is that the overall process probably got contributions from both sources, so I don't think you should call them competing.
You published something a few years ago where you found that, I'm going to quote from an article I read about your paper, you found long RNA molecules form when nucleoside triphosphates, in other words, building blocks of life, do nothing more than percolate through basaltic glass.
This was a very surprising discovery for us. We did incubate nucleotide triphosphates with volcanic glasses, so basaltic glasses, and we found that these were producing high molecular weight products that did seem to be RNA. And then we went through a couple of years and 60 pages of supplementary material and chemical probing and analysis and testing. And we found that these molecules, these high molecular weight products, contained phosphodiester linkages, which are the bonds that keep together building blocks of RNA, so the nucleotides, which basically defines RNA itself. So yes, we think that we have found a pretty straightforward pathway towards the oligomerization of RNA in prebiotic conditions. And this happens, it's aided by surfaces that were present in the Hadean, were widespread. It's a pretty general process, so you do not have to pick a specific location and a very specific point where this might have happened. So these might have happened all over the crust of Earth, well, the crust that was present at the time, and in other planets too, because these are surfaces that you can find.
The crust of the Earth at the time, you mean ancient Earth?
Yes, the Hadean Aeon.
We found that there was basaltic glass was everywhere.
This was produced both by volcanism and by impacts, actually, with asteroids from outside. Mm-hmm. So the impact would produce the basaltic glasses that we're using for our experiments.
So from the public's point of view, they understand building blocks of life. I mean, the ingredients, you mix them together, and then out pops something that walks around maybe one day, eventually,
one day,
right?
Eventually.
You take these ingredients, You mix in some basaltic glass, give it some time, and then eventually RNA forms. And now we know RNA. RNA is essentially just half of DNA.
RNA as a single-stranded molecule can actually fold into itself and create double helices just like DNA. So the point of RNA being a more interesting, if you wish, molecule than DNA is it does have an extra reactive group that is the OH on the 2' of the sugar. Without being too detailed, this provides more reactivity to RNA, and that is why we know for a fact that RNA is capable of both preserving genetic information with mutations, which defines Darwinian evolution, and also is capable of catalytic activities that are now mostly relegated to proteins in our extant biology.
Are there life forms today that do not have DNA, that only have RNA?
Well, yeah, there's viruses and other things, but RNA in certain specific cases, and in the case that I'm talking about, the ribosome, is actually performing the functions that are otherwise relegated to proteins. So you can see how in a prebiotic environment where molecules are trying to establish themselves and there's really no cells yet, there's no organisms, one molecule might have tried at the time to take on all the work. So I need to replicate myself. I need to establish myself in this environment. How am I going to do it? I need to both maintain enough information to then be able to pass it to the next generation, and I need to be able to do that myself, to effectuate it myself. So do the job, you know.
This chemical soup, right?
Yeah.
That can form RNA just through a natural process.
Yeah.
This is significant because it starts to look like the precursor to life.
There is a point in molecular evolution that we tend to call the genetic takeover, where, and this is my view of the genetic takeover. So at a point when the atmosphere and your environment is stable enough, you can actually, besides starting, after thinking about trying to survive and trying to evolve enough functions for your survival, then you can start relaxing, so to speak, and thinking about storage of information and about getting more stable, so that then you can evolve toward more fancy functions or forms. And that is when DNA came around. DNA is the stronger, more stable molecule that can just sit there and hold on to the information that is needed without having to do any extra job. So focusing just on the storage of the information. And that is kind of when RNA, you know, in this very simplified story about the genetic takeover from RNA to DNA and to protein, so that the biology that we know now, that is how RNA would have just moved into, okay, I'll give you the function to proteins and I'll give you the information to DNA.
Does this help contribute to the notion that life could have emerged here on Earth independently just through a natural process?
Oh, absolutely, yes.
This provides strong evidence for that.
Well, yeah, that's what we hope. We have shown that there are processes, easier processes than all the crazy chemistry that sometimes we try to do in the lab and we try to force in the lab. Mm-hmm. There are easier processes that might have actually lead to the polymerization of the first building blocks, and then, you know, towards evolution. Of course, we are still trying to understand what these products that we are observing can do and cannot do, how much of an established molecule they might be. We're still looking into all of that and the details of and the mechanisms of the formation. But absolutely, yes, this is strong evidence that there are processes that don't need to be so complex.
You're saying this is a much more simplified process, a natural process, and provides an easier answer to this question of how life could have arose independently.
Yeah, absolutely. So this is a simplified process in the sense that we did not have to go through crazy hoops to observe. But what we have done is an observation. Instead of just mixing and pouring and boiling, we just observed these happening. So that is definitely a major step forward into trying to understand what prebiotic chemistry can give us, really. But on the other hand, we don't know how this is happening, and by the time we go through all the details and we figure out how this is happening, I wouldn't call prebiotic chemistry extremely simple. It's probably complex itself. It's just it shouldn't be you who adds the complexity. Mm-hmm. You should just be observing the complexity that nature went through.
Mm-hmm. I don't think most people have heard of this. They know we're trying to...
Oh, they do.
Maybe in your community. I'm talking about the public.
Oh, I see.
You publish research which you find quite compelling, otherwise you wouldn't put your name on it.
Correct.
You think there's very strong evidence
Correct.
for this process to have occurred. It's reproducible. You can observe it more than just once.
Oh yeah, a million times. We did this a million times.
A million times.
We're doing it now. I should show you some of them.
You should show it to me.
Yes.
Okay, but there are some in your community who are skeptical.
Yes.
What's that like?
We have RNA, and I don't know how else to say it to not piss off people, excuse the French.
In this article that was written about your paper, what was highlighted is that it's relevant to Mars because we think Mars had the same conditions of early Earth.
Yes, yes. And that's why I was saying, that's why it's such a fascinating process to discover, because it's really applicable to Mars and most likely, not potentially, most likely to many, many other planets.
The idea that Mars could have developed RNA in the same way that early Earth could have is not so crazy.
It is not, definitely. If you consider also, again, like when you look at things in relative terms, Mars is so close to us. It's our own solar system. It is very well conceivable that it would have had a molecular evolution just as we did.
But if we do find life elsewhere, and we do find that it's common, some think that's going to be a blow to our ego. What I'm hearing you say is that that's fine, we should still look for life anyway, even if that's the outcome.
To be honest, I don't think that it is even possible, like given the numbers of the universe, the number of stars, the number of solar systems. This is very basic talking about this, but thinking that we are unique, it should be a given that we're not special.
This is the continuing series of Blue Planet Red, an award-winning documentary film on the history, evidence for life, and catastrophes that contributed to the death of Mars. The documentary includes interviews with NASA's lead astrobiologist, university professors, aerospace industry scientists, and independent researchers. If Mars was like Earth in the distant past, could there still be life there now? What happened to it? And what turned a blue planet red?